Every cell comes to a point where it has reached a certain size and usually stops growing or dividing. The cell cycle is the stages in which cells grow, replicate, or divide. This process of dividing can take place in 8 to 20 hours. The two main phases of the cell cycle are interphase and M-phase. A eukaryotic cell spends most of its time in interphase. The first part of the interphase is the G1 phase, a gap where DNA synthesis does not occur. In this stage, the cell grows and normal metabolism occurs, but the cell does not divide. Also during this phase, the enzymes that are necessary for DNA synthesis become active and allow the cell to continue onto the S phase. In the S phase, or synthesis phase, DNA goes through a replication and histone proteins become synthesized in order that the cell can produce duplicates of the chromosomes. The last phase is G2, a shorter gap phase in which protein synthesis continues, making the cell close to division. The M phase consists of mitosis and cytokinesis. The purpose of mitosis is to produce new cells for growth, repair, and development. During this process, mitosis separates sister chromatids, complete sets of chromosomes at opposite ends of cell, so each daughter cell receives a complete set of chromosomes with exactly the same genes as the original cell.
Prophase. During this first mitosis stage, the long chromatin fibers, which are replicated DNA, compact through a coiling process that makes them decrease in size but become more condensed. Each replicated chromosomes are comprised of two sister chromatids, both with the same genetic information. These sister chromatids are linked by cohesion, a ring-shaped protein complex that helps to maintain accurate chromosome segregation. All chromatids have a constricted section, called the centromere, in which the sister chromatids are bound. Cateinophores, a multi-protein complex, are attached to the centromeres. This allows microtubules, hollow cylindrical fibers, to bind. These microtubules are responsible for cell shape, motility, attachment to other cells, and in chromosome distribution. We can imagine a dividing cell as a globe. The microtubules spread out their protein fibers from the poles as they form the mitotic spindle. The mitotic spindle serves to separate the duplicated chromosomes during anaphase.
Here, the long chromatin fibers are coiling. The identical sister chromatids are now visible. The centromere, depicted by the purple gloves, holds the chromatids together. Their kinetochores, the blue gloves, are also apparent.
Prometaphase. In this stage, the nuclear envelope breaks down, so there is no longer a recognizable nucleus. As the envelope breaks, the duplicated chromosomes are spread throughout the region. Some mitotic spindle fibers elongate from the centrosomes and attach to kinetochores. Other spindle fibers elongate, but instead of attaching to chromosomes, overlap each other at the cell center. The kinetochore microtubules attach to the kinetochore proteins of the sister chromatids during prometaphase.
In metaphase, tension applied by the spindle fibers aligns all of the chromosomes in one plane at the center of the cell, known as the metaphase plate. Each chromatid has been fully condensed and appears to be distinct. Since the chromosomes are clearly visible during this metaphase, the chromosome composition, karyotype, is observed for abnormalities. There are 3 types of microtubules found in the meiotic spindle: polar, kinetochore, and astral. Polar microtubules elongate from the pole to the middle region, at which they overlap and intermingle with the polar tubules from the other side of the pole. Kinetochores extend from each pole to join the chromosomes at their kinetochores. Astral microtubules are the shorter tubules, forming asters at each pole. The sister chromatids are now pulled into alignment with the metaphase plate. In this step, the sister chromatids begin to separate. As they separate, they are no longer called chromatids but chromosomes. These chromosomes, with the help of spindle microtubules, begin to move towards the opposite poles. Research is still being conducted on how chromosomes actually move through the cell. Some biologists say that the shortening mechanism of microtubules causes the chromosome to pull towards their poles. Others say that since the microtubules slide, decreasing the overlap when they elongate, the chromosomes are indirectly moved apart. They are attached to the poles by the kinetophores. The sister chromatids separate, somewhat unwillingly in this video, and move toward the poles. Telophase. This is the last stage. The daughter chromosomes arrive at the poles and the spindle fibers that have pulled them apart disappear. The nuclei are rearranged. The chromosomes loosen and partially uncoil as a new nuclear envelope made from vesicles and other parts of the old envelope forms around the chromosomes.